Hello, I'm Naveen and my presentation is going to be about red yeast fermentation and growth. Here we have the outline of the presentation where we'll have the introduction, materials and methods, results and discussion, conclusion, acknowledgement, and finally references. This experiment involves red yeast. Red yeast rice is a fermented type of rice produced using a species of mold known as Monascus purpureus. This rice has often been used in traditional Chinese medicine and was found to have the compound monocolin K. Monocolin K is often found in cholesterol lowering medicines. In this experiment, we plan to take this red yeast rice and then to isolate it, ferment, and then grow it in the laboratory. Under methods, the first task is to prepare media for isolation and fermentation. Afterwards, we move on to the isolation of microbes. In this case, we isolate red yeast. Then we establish a pure culture of the isolated microbe. Afterwards, we proceed with fermentation and inoculation. And finally, we move on to growth kinetics and yield analysis. The isolation of red yeast was carried out by first grounding a sample of red yeast into a powder and diluting it by a serial dilution of 5 or 10-fold. Then 50 microliters of the diluted sample was transferred aseptically onto an agar plate and spread using a sterile spreader. Finally, the plate was left to incubate at a room temperature for about a week. Under fermentation, we first measure and label the weights of each of the 9 pieces of 50 milliliter Falcon tubes with their caps on. Then we transfer 20 milliliter of potato dextrose broth into each of these Falcon tubes aseptically. One of these Falcon tubes is kept as a control, and then one plug of agar containing one mature colony of red yeast was transferred into each of the remaining tubes for inoculation. One of these inoculated tubes was kept under 4 degrees Celsius as the time zero sample. The caps were closed slightly loose for all the replicates and then taped with masking tape. Finally, the tubes were incubated at 20 degrees Celsius in a rotary shaker for 8 days. Under yield analysis, we first collect the Falcon tubes containing the pigment at different times, and then, using a spectrophotometer, we measure the OD of the pigment colors, where we measure yellow at 375 Newton meter, orange at 475 Newton meter, and red at 500 Newton meter. 1 milliliter of the supernatant was collected and measured at their respective wavelengths for these OD measurements. Here we have visual results of the red yeast growth on an agar plate at its initial growth phase and its growth after 7 days. During its initial growth phase, we can see there are only slight light orange pigmentations on few spots on the agar plate. However, after 7 days we can see that there are dark orange or red-brown colored pigments all over the surface of the agar plate. Unfortunately, Due to problems encountered and failure to collect proper data, the growth rate and results of the yield analysis of this experiment were not able to be calculated. Through the obtained results of this experiment, the proper growth of red yeast and potato dextrose agar can be visually confirmed. However, numerical calculations and the graphical interpretations of the growth rate and results of yield analysis are required to go further. Special thanks to Dr. Daniel Stanley Tang for the weekly practical sessions and emails sent to us notifying us about the status of the practical, and also for helping the online students who weren't able to come for the on-campus lab sessions. Here are the references, and thus we have reached the end of the presentation. Thank you for your time.